Okay, boys. Once again, good morning to all of you. We are discussing an important chapter, molecular inheritance of the cell. And this chapter, as I told you, is not only important for the sake of your board exams, it builds up your career. Who knows, some of you may become in future molecular biologists and you may work under Nobel laureates. So this gives you a career in future. And third most important thing is, it will help us to know what is inside us and why we are what we are today and why we will be what we will be in future, right? So many questions that arise in our mind will be answered through this chapter. So basically, so far we have cleared the doubt that we had in our mind. What is the material that is getting inherited? We had doubt. Is it the chromosomes? Is it the nucleus? Is it the DNA? Is it the proteins? Like that. So far, with the experiments that have been done by scientists like Hershey Chase, so without any doubt, we have concluded that DNA is the genetic material. Further, we improve our knowledge, wherein for many years the question was there that is it the DNA that is getting inherited or something else is there? Whatever characters that we are showing physically, physiologically, morphologically, is it because of DNA or something else? So these questions started arising. And thanks to this gentleman, Professor Crick, and he proposed the central dogma of molecular biology. What is that central dogma? We can say the secret of our life. The secret of our life is DNA undergoes replication. We call it DNA replication. So two vertical threads will be broken vertically and form a single thread. That single thread is RNA. And RNA will ultimately form proteins through a process called translation. This is only the crisp of the matter. We are only talking about the headings. And in future classes, we will be describing what are the sub-steps that will be there in transcription and translation. Okay? Overall picture we are talking about today. So that we will not have doubt in our minds that the secret of our life is proteins. So first we thought of it as nucleus. Then, of course, to begin with, transforming principle. What did Griffith say? Something has transformed, isn't it? Remember S strain and R strain? R strain transformed to S strain because of something, he said. Now, later on, people thought it must be nucleus. Further experiments, they thought it is the chromosomes. Then comes DNA, then comes RNA. Now we have arrived at the highest step, proteins. Proteins are our secrets. How these proteins are formed? With the help of DNA. So DNA, because of weak hydrogen bonds, will be broken vertically. The bond between the nitrogen bases will be broken. As a result, two threads will be formed and each thread would be like RNA. So DNA forms RNA due to replication. DNA replication means breaking of DNA. And RNA, through a process called translation, forms proteins. And proteins are our secrets. Whatever proteins that are there in our body, they are responsible for the characters, traits that we display. And as Sandeep said yesterday, there is something called reverse transcriptase in viruses, because in viruses, there are viruses, I said, wherein RNA will be the genetic material. So the single thread undergoes duplication, forms DNA. This RNA forming DNA is called reverse transcription. Why is it so, Sandeep? Transcription is DNA forming RNA. DNA forming RNA is transcription. Now it is becoming ulta. RNA is forming DNA. Hence Sandeep calls it reverse transcription. And as we said, with the help of two steps, one transcription, second translation, proteins will be synthesized. So first step, that is transcription. For transcription to take place, what is essential? DNA replication is a must. So replication is the copying of DNA from parental DNA. Let me expand that. So Professor Watson and Professor Craig proposed semi-conservative model of replication. Now I'll repeat the word semi-conservative model. What do you mean by conservative? Some item is there in Prasad's house. Maybe it is coming from his grandparents, then grandparents, then parents, then Prasad children. So that item is still there. Then we say conservative. We conserve, we preserve. Here what is that? Semi-conservative, half preserved. Out of two threads of DNA, one thread is the parental thread. That is what we mean when we say semi-conservative. Okay? So always the Professor Crick and Watson proposed that the DNA replication must be through semi-conservative method. It suggests that the DNA strands act as template. Template means source for the synthesis of new complementary strands. After replication, each DNA molecule would have one parental and one new strand. Let us see this. Let me expand that. First, all of you appreciate these two gentlemen, Professor Watson and Professor Craig, and focus on parental DNA, top, and the parental DNA. Sandeep is putting some small Benkias on Sandeep. The moment he supplies a bit of energy, hydrogen bond is broken. So two vertical threads will be separated. And remember Prasad, these two, the blue ones, blue ribbon, you focus. All of you know that both the two blue ribbons are parental strands, correct? And the parental strand, blue ribbon, consists of, already I said, SP, superentopolase, sugar plus phosphate. And to that attached is nitrogenous bases. So only Kadkol said, what is DNA? DNA is equal to SPN, sugar, phosphate, nitrogenous bases. Correct, no Kadkol? And also we talked about base pairing. One purine pairs with pyrimidine and vice versa. Pyrimidine pairs with purine. So in the parental strand, let us say there is adenine. So in the new strand that gets synthesized, what would be the base pair for adenine? Thymine. Remember we said AT and TA ta. So it has to be either AT or TA always. So adenine thymine or thymine adenine. So if the parental strand, that blue ribbon contains, let us say, adenine, the newly synthesized daughter DNA part or newly synthesized portion of DNA will be having thymine. Suppose the blue ribbon has got guanine, parental DNA strand. What would be the base pair the other side? Newly synthesized one? Yes, Kadkol? Cytosine. And vice versa. It can be cytosine and guanine. So like this, since one of the parental strands is part of the new daughter DNA, Professor Watson and Crick would like to call it as semi-conservative, because one strand is there from the parent side. Another strand is the new strand. He calls it, see, old strands, parental strands, new strands. So totally daughter DNA is formed. Right? That's why they said DNA replication is always through semi-conservative method. And we must salute this man, these two gentlemen also, Michelson and Stahl. They performed one experiment. What is that experiment? To prove that, yes, Michelson and Stahl's experiment. Look at that. And again, lot of chemistry is involved, Kadkol, in this. All of you know that nitrogenous bases will have nitrogen as the main component. Correct, no? And you know that nitrogen also can be an isotope. So what is that he has done? Normal one, as all of you know, is N714. What do you say, Kadkol? Atomic number and atomic mass, right? 7 and 14. Am I correct, Sandeep?
Yes.
Yeah. And Rakesh, when it is put 15, इसका मतलब क्या है? N15. हाँ जी.
It's
हाँ, atomic mass is 15. इसका मतलब क्या एक्स्ट्रा ऐड हो गया? हाँ? Yes, Rakesh? Neutron. Because neutron is heavy, isn't it? Without any charge. Hence, when we write N715, 7 is the atomic number, means 7 electrons or 7 protons, 7 plus charges, 7 minus charges. And that becomes 14, but when we write 15, one neutron is added. So the charge is still balanced, 7 plus 7 minus. Only thing is, one neutron is added, right? Now, they measured the density, right? In one strand, the Michelson and Stahl. Now, let me read once, then you will have an understanding. So, they prepared 2 culture media of E. coli. What is E. coli, Sandeep? Escherichia coli. It's a bacteria found in the stomach and intestine of Sandeep. It's a good friend of Sandeep, just like Kadkol. Okay? One preparation contains NH4Cl. NH4Cl means? Ammonium chloride. So, in that, look at that, what they have done, Michelson and Stahl, instead of normal nitrogen, what they have done? They incorporated isotope of nitrogen. N15 they have used. Isotope of nitrogen. So, N15 was incorporated into both strands of bacterial DNA and the DNA became heavier naturally. Then, other preparation contains normal one, N14, means 7 plus 7 minus, 7 electrons, 7 protons. So, N14 was incorporated in both the strands and the DNA became lighter. That means, what Michelson and Stahl they have done? They have prepared a heavier DNA using N15 isotope of nitrogen, another normal one, lighter one. So, these 2 types of DNA can be separated by centrifugal mission. In that, they have used one sismium chloride, CSCl density gradient. Okay? Now, look at this. This is what they have done. And I am sure all of you must have guessed why these 2 gentlemen have done this preparation. Any idea? Sandeep? Prasad? What is the purpose of this experiment? Any idea? Yes, Prasad, you are not audible. Speak loudly. Okay. Kadkol?
DNA
DNA is the genetic material. That we have proved now already, Kadko. Already we have proved beyond doubt. DNA is the genetic material. Transforming principle followed by the 3 McLeod experiment. Why Michelson and Stahl did this experiment again? Any? Rakesh Mohanty, Jai Jagannath. Come on, Rakesh. राकेश सोच रहे सर काम ही नहीं है इन लोगों को, कुछ ना कुछ करते रहते हैं. एंड दैट कम्स इन आवर टेक्स्ट बुक. या संदीप. Yes, Sandeep? Yeah. See, what is the proposal of, yes, Sandeep? What is the proposal of Professor Crick and Watson? Semi-conservative, they said. And when we say semi-conservative, what is that we have said? One of the strands in the daughter DNA, let me show you again. See, look at that. Blue ribbon I said. One of the strands in the daughter DNA is parental DNA. New one is the one red color one is the new one. So the daughter DNA is a combination of new thread, red ribbon, and the blue ribbon, old one. So the intention of Michelson and Stahl is to use that. In one ribbon, nitrogen bases will be N14. In another, it will be N15. So if the daughter DNA gets both the strands, one strand containing N14, another strand containing N15, Professor Crick's experiment is proved. So basically, Michelson and Stahl performed this pardon? Both cannot be new. You are right. Both cannot be new. But under some conditions it can be new. Okay? That also is not impossible. You must believe in science, that in science nothing is impossible. You can do it. Right? But only some conditions are required. Some facilities are to be there. Then we can do it. So it's the will of the scientist, ultimately. So the purpose, I hope you have understood. The purpose is to prove that new strand will have that N14, the old one will have N15 or vice versa. Overall, the daughter DNA will have 2 strands, one old parental strand, one new DNA strand. So they used N14 and N15. And since it is heavier, heavy one will be at the bottom, lighter one will be up. Density gradient you can see, and the medium they have used is caesium chloride, CsCl. See that N15 being heavier is at the bottom after the centrifugation. N14 is lighter one at the top. So this is what they have done. And further, let me show you that yes, 2 types of DNA can be separated by, I think we have covered, centrifugation in a CsCl density gradient. Now, what is that we have? They took E. coli cells from N15 medium and transferred to N14 medium. And after one generation, that is, time period is 20 minutes. After 20 minutes, they isolated and centrifuged the DNA. Now, its density was intermediate hybrid because it is N15 and N14. One strand is made up of normal nitrogen. Another strand is made up of heavy dose of nitrogen, isotope of nitrogen. So since it is a hybrid, the weight is neither too high nor too low. Again they did centrifugation, and you can see this. This shows in newly formed DNA, one strand is old, that is isotope 1, and one strand is new. This confirms semi-conservative replication of DNA. Now same thing here you can see. Let me expand. Yes, you can see. Look at that. There is a heavy dose. Parental one basically is incorporated with nitrogen 15, isotope of nitrogen. After 20 minutes, it is broken. right? Replication has taken place. And new strand, here it is shown with green color, N14, and old one, heavy, N15. After 40 minutes, what happened? Look at that. Let me show you a little bit up. Yes, you can see now. Generation 2, N14, both the strands, and 2 strands is a hybrid one, having one strand N15, another strand N14, and the last one again is the N14 strand. And you can see in the centrifugal machine the concentration. Light ones are up, N14, N14, hybrid one is in the middle, and let us see the N15, both the strands. I am sure it will be at the bottom. Look at that. Red color, heavy, the parental one. So this is how Michelson and Stahl proved that the daughter cells, daughter DNAs that are formed will be through semi-conservative method. Okay? And then comes one more experiment. Taylor and colleagues in 1958, you see, slowly we are coming, performed similar experiments on the faba beans, see a faba, the beans that we are eating as our palle, okay, sambar, all this, using radioactive thymidine. Remember T for thymidine, to detect the distribution of newly synthesized DNA in the chromosomes. And he proved that DNA in chromosomes also replicate semi-conservatively. This almost clue is given by Michelson and Stahl. Look at that, chromatids and thymidine, and labeled thymidine, that is isotope, and semi-conservation is seen. Okay, this is done. And then what are the things required? Is it so easy that as we speak the DNA simply will break off just by applying or supplying energy? Hydrogen bonds are broken and the DNA gets replicated. It's not so easy. We need many enzymes for this. Enzymes play a huge role in this. So DNA replication starts at a point called ORI. ORI is origin of replication. Look at that in the diagram. So there is a DNA ladder. In the ladder, at particular point, breaking of DNA is taking place. The point at which the DNA strands are breaking is called ORI, origin of replication. It is not that anywhere it can stand. At any place it can stand. No, only at ORI places it will stand. A unit of replication with one origin is called replicon. During replication, the two strands unwind and separate by breaking hydrogen bonds in the presence of an enzyme helicase. This enzyme is important to break hydrogen bonds. Unwinding of DNA molecule at a point forms a Y-shaped structure called replication fork. Let me show you that. Yeah, look at that. Yeah. First, focus on ORI, origin of replication. You can see the breaking of the strands. Anurag is waiting in the lobby. Okay, why Anurag is waiting still? Let's come now. Okay. Hey. Prasad was here, no? Right. So, first and foremost is the place, what we call ORI. Step number 1 is initiation of replication at a particular place called ORI. Second, bidirectional replication at one origin, that is both the sides it is breaking. Bidirectional replication continues until terminal is reached, end position is reached. This continuation. But what you must remember here is ORI. It is not that any place at any time this DNA will be broken. There has to be a ORI. And once this ORI continues, the process of breaking continues, what we said, Y-shaped structure will be formed because the thread gets unwinding, and that Y-shaped structure is called replication fork. You can see here, replication fork. So one side it is unwinding. Focus on the brown color one. Brown color one is the parental strand, both up and down. Blue color one is the new strand. So it appears like Y-shaped, and that Y-shaped structure is called replication fork. So the separated strands act as templates for the synthesis of new strands, and always the replication is 5' to 3'. Look at that. Replication of DNA is from 5' to 3' direction. On one strand you can see the fifth carbon, right? When we say 5', 3', what does it mean? The carbon number to which it is attached. Remember, sugar is attached to phosphate, phosphate is attached to nitrogen bases. So when we say 5', 3', it is the number of carbon. So the DNA gets replicated from 5' to 3'. That is the direction. And then deoxyribonucleoside triphosphates. Okay, what do you mean by this? Deoxyribonucleoside. Remember, nucleoside, I said. Two things we were talking: nucleoside, nucleotide. Nucleoside, N, S. I said in the name only clue is there. N for nitrogenous base, S for sugar. Sugar plus nitrogenous base is the nucleoside. To that, phosphate is added. Then that provide energy for polymerization. See, to break or to make, energy is required. So when we are breaking the DNA molecule, who will give the energy for that? These are the chemical substances that give energy. So they are called DATP, DGTP. What do you mean by DATP? Adenosine triphosphate, right? Guanine triphosphate, cytosine triphosphate, thymidine triphosphate. So these are the substances that provide energy. Okay. Now look at that replication fork.
Sir, why D, sir?
D is the source of energy, deoxy. We said DNA, sugar molecule, what is it? Deoxyribulose. The D stands for deoxyribulose, right? And now you can see here how the DNA molecule is being broken. Replication fork, I am expanding it. Look at that. And look at the role of helicase, how it is breaking the bonds, hydrogen bonds. And a Japanese scientist observed that on one side it will be a continuous strand, on another side it will be broken ones. So he has named it as leading strand and lagging strand. Okay, leading strand, it's a continuous one. Lagging strand, breaks are there in between, broken strands, right? So this DNA synthesis, particularly daughter DNA synthesis, requires polymerization. And second, parental DNA to be broken requires energy. So energy availability is a must for both. And who is providing that? As we said, this. You can see the replication fork formation and all these DNA molecules, where are they? They are inside the chromosome. And just to differentiate, he is giving colors: adenine, thymine, cytosine, guanine. So basically, this is how the DNA replication takes place. Now, first we said two things are required for DNA replication. One is the presence of helicase, an enzyme that breaks the hydrogen bonds, and the breaking will be in a place called ORI, origin of replication. And then we said that there has to be energy giving substances. They are called deoxyribonucleoside triphosphates like DATP, DGTP, so that polymerization of DNA takes place and new daughter DNAs will be synthesized. Right? Clear? So far, three things are required. I hope you are following me.
Yes, sir.
Yeah. Next, we require what is called small portion of RNA primer. So, firstly, a small RNA primer is synthesized in the presence of an enzyme called primase. This is the second enzyme that is required. Primase is an enzyme that synthesizes RNA primer. And in the presence of an enzyme, DNA dependent DNA polymerase, many nucleotides join with one another to primer strand and form a polynucleotide chain. Okay. Now the second one, let me expand. Yeah. So we have got the leading strand and lagging strand. Now look at that. Brown one is the parental strand. I hope you are clear. Focus on the brown thread, up and down, up 3 prime, down 5 prime. And the daughter strand, blue ribbon, is being synthesized. So for that, what is required? RNA primer, right? And if you carefully observe, top one we said is a continuous chain. The blue ribbon is continuous, whereas at the bottom, see, the blue ribbon is broken. And I said one Japanese scientist first discovered this. So his name was Okazaki. So these little bit pieces are named after the scientist as Okazaki fragments. They are discontinuous thread. Okay? And synthesis of lagging strand is taking place, synthesis of leading strand also is taking place. So as a result, you are getting 2 daughter cells. And then look at that. Parental DNA. Now role of primase. RNA primer you see. Yes. So because of the presence of that, the new daughter cells are being synthesized. One is the continuously synthesis is taking place. We call that as leading strand. Another is with the pieces. That is lagging strand because of presence of Okazaki fragments. So now first we said enzyme helicase is required so that hydrogen bonds are broken. Second we said the breaking will take place at particular place in DNA called ORI, origin of replication. Correct? And third we said that there should be energy providing chemicals for the formation of new daughter DNA. We said polymerization of nucleotides. What is polymerization of nucleotides? DNA, new DNA formation. And then fourth we said there should be a small RNA primer which will initiate the formation of new daughter cells. And there will be 2 types of strands that will be formed. One is the continuous synthesis, that's called leading strand. Another is the strand with broken parts, Wakajaki fragments, that is lagging strand, right? I think we will continue further tomorrow to talk about Wakajaki fragments. Until then, keep studying with a focus, right? Take care.